Hello, Sp2021 students. Today we're going to go over Chapter 15, Antimicrobial Drugs. With this chapter, I've kind of condensed it to the really important parts. So make sure you're looking over the PowerPoint and watching this video to get the key information. And when you go to look at it in the book, remember, you're only responsible for what I talk about or if I tell you what to focus on in the book. Now, we're going to start getting into, after this chapter, diseases of the organ systems. We'll get into the digestive, respiratory, nervous, integumentary, all the ones you can really think of. We'll get into those and talk about specific diseases within each of those organ systems. But first, I want to talk a little bit about antibiotics because they're really important. And I've seen a lot of people and heard a lot of people asking about antibiotics and COVID-19, which, being a virus, it doesn't work. Antibiotics won't work for viruses. We have to use antivirals. But we'll talk about that in a little bit. All right, so Chapter 15. Antimicrobial drugs are basically these compounds that kill or inhibit the growth of uh, bacteria, or if you're looking at an antibiotic. If you're looking at an antiviral virus or an fungal, it'll suppress fungal growth or kill it. Now, this is really the antimicrobial drugs, knowing the fact that they can be killed or inhibited is really important, because in some cases you don't want to kill the pathogen, because then it makes the situation much, much, much worse. And I have a really good example of that in a few minutes that I will talk about. But there are antimicrobial drugs that you take. Let's say if you have a bacterial infection, you take an antibiotic. That antibiotic has a very specific target, and it goes and targets that specific thing within a bacterial cell and kills it. Now, what we call bacteriostatic or inhibiting growth, is another effective way to uh, limit and or stop an infection. If we give a patient antibiotics that trigger this inhibition of growth for the pathogen, it allows the immune system to catch up and really take control and be able to fight that pathogen. All right, so antibacterial is our antibiotics, antiviral drugs against viruses, and antifungal go against fungal diseases. Excuse me. Um, So before antimicrobial drugs, people died from the most simple things things that we consider simple infections, like strep throat or a simple wound from being in the mud and in war. It gets infected because it's not cleaned properly. They're dragging themselves through the mud and dirt, and it gets infected. Because also one of the things you should keep in mind, right, is Clostridium tetani the endospores live in the soil. So it's possible to get infections in that manner. Before antimicrobial drugs, something like strep throat could kill somebody. For us, strep throat is an inconvenience, and we take antibiotics, and we're usually good. Now, granted, obviously there are other cases where it's not that easy, but for the most part, strep throat is not something that we're overly concerned with nowadays. Um, Fleming. So Fleming was this scientist who actually discovered the first antibiotic, penicillin. What he was doing was he had a plate, an agar plate, and what he was trying to grow was a Staphylococci species. I don't remember which one at the moment, but it was definitely a Staphylococci species. He was trying to grow it on that plate. And on accident, this fungal spore lands on the plate and it grows. And it grew and grew and grew. And what Fleming noticed is near where the fungal colony was, there was absolutely no bacterial growth, which told him, well, that that the fungus or the penicillin, it was a mold, the penicillium mold, was secreting some chemical that was inhibiting the growth of that pathogen near it. So it secreted that chemical, that compound out, and then no pathogen, no Staphylococci species could survive next to it. So penicillin was on accident. He discovered it on accident. It was a happy accident, obviously, but it was definitely an accident. And now, well, actually, let me correct myself and say, for a long time after that, penicillin was the gold standard for antibiotics. But nowadays, we actually see lots and lots of resistance to penicillin. And then most recently, within the last 10 years or so, we're starting to see resistance of penicillin drop in pathogens. So that's kind of interesting. We have the penicillin became really resistant, the bacteria became really resistant to penicillin after a while, and then we switched to other antibiotics, and the amount of resistance to penicillin actually dropped. So it's very interesting to see how these antibiotics can fluctuate in their effectiveness. All right, but Fleming accidentally discovered it, and he's credited with discovering penicillin. Two types of extents of which these drugs work: broad spectrum versus narrow spectrum. Broad spectrum is just exactly what it sounds like. It attacks a wide variety of organisms, whether it's gram positive or gram negative or other atypical uh, pathogens, so in terms of their cell wall, because penicillin actually, for instance, penicillin attacks cell walls. So if you have an atypical cell wall, or if you're gram positive, gram negative, you would want to use a broad spectrum antibiotic. But when you get really down to it and you say, okay, well, this patient has this pathogen X, let's say it is Helicobacter pylori or E. coli 0157H7. You know exactly what's causing it and where that would be located. You give a more narrow spectrum antibiotic, which is essentially more targeted. So when you give a broad spectrum, it essentially throws a blanket over all the pathogens and all the cells that have the trait it's looking for, whether they're bad or good cells. They don't differentiate, and they'll just destroy them all. Where narrow spectrum have a very specific target that, if the cell doesn't have that target, then it's going to survive. So you use narrow spectrum in very specific cases and with certain pathogens. You don't want to just blanket cover them with a narrow spectrum antibiotic. You want to use more of a broad spectrum antibiotic. And I kind of mentioned these earlier: bacteriostatic versus bactericidal. Static means inhibit, cidal means kill. So any antibiotics that are bacteriostatic inhibit the growth of the pathogen, or pathogens. Bactericidal will actually kill the bacterial pathogen or pathogens, because again, static versus killing. Now, there is a right time and a right place to use each one. They are not something that you want to just throw around without taking a look at what you're giving, because let's say you have a gram-negative cell that produces an endotoxin. We should know from the last chapter, from uh, the chapter about toxins, that the LPS, which is part of the outer membrane of the gram-negative, is where the endotoxin is found. Now, if you give a person a bactericidal antibiotic and it kills that cell and all the other ones that are uh, disease causing, all the other pathogens, it's going to release a large number of endotoxins into the body, and the patient can actually get much, much worse in terms of health. They can get much sicker. And that's because, yes, there's an infection. Yes, an infection is bad. But once you give that antibiotic, that bactericidal antibiotic, it kills all those gram-negative pathogens, if that's what they're targeting. And what'll end up happening is the LPS breaks apart and it gets all the endotoxins are secreted into the lymphatic and bloodstream, and the patient gets much worse. So there are times and places, specific times and places, for bacteriostatic versus bactericidal. Um, so drugs' properties can be changed based on different things. So the pathogen type itself, whether it's an enteric, meaning living in the guts, or it's a respiratory, or an aerobe, living in the lungs, dosage, length of dosage, the amount of the pathogen, and then the route of administration. So two of these that I really want to talk about quick are pathogen load, okay, pathogen load, uh, and the length of drug regimen. So pathogen load is essentially how many pathogens are present. And we've talked before about how Shigella, for instance, only requires less than 20 cells to cause an infection. Okay, so by having less than 20 cells, that pathogen load is pretty low. However, since it is so pathogenic, it only needs that few cells, those few cells. So the pathogen load may be low, but the organism is so pathogenic that it's still effective. Whereas E. coli, for instance, E. coli 0157H7, uh, it takes a lot of cells for them to cause an infection. So the number of pathogens will absolutely affect the antibiotic that you're giving, in, if we're using bacteria as an example, which we are. Uh, so the pathogen load is going to be something to keep in mind. Now, if it's early on in the infection, you'd expect that the pathogen load would be pretty low. But once it reaches that logarithmic stage, right, the next stage in the growth, then you're getting extremely high numbers of pathogens that are found in that host, or excuse me, in that host. And then the length of drug regimen. Many of you have probably been given antibiotics for some infection at some point in your life, and it tells you to take this antibiotic this many times a day for this many days. And I guarantee you, some of you out there, which you may not admit it or not, I'll admit it, I'm guilty of that in the past, where after about 5 days, I start to feel a lot better, and I'm like, okay, I'm done. I don't need these antibiotics anymore, and I don't finish the regimen. This is a problem because what you need to do is you need to get a high enough concentration of the antibiotic in your body to be effective, right? And if you don't have that concentration high enough, sure, it may kill a good chunk of the pathogens, but those that are there will become resistant, or they could become resistant at least. So it's always important to take all the antibiotics for the entire regimen, because by condensing or shortening the length you take them for, that becomes a big problem in the end, and that's where antibiotic resistance is starting to develop. Really, when it comes down to it, antibiotic resistance is the cause of human misuse of antibiotics. We are the cause of it. We're the ones that are causing the problem. Um, no one likes to hear that, but um, it's the truth. So we'll get back to that in a little bit. Okay, so let's move on. Uh, there are, again, to bactericidal versus bacteriostatic, this argument I've already kind of made. Um, Bactericidal may seem better to use, but in certain cases, it's important that we look at it carefully and decide which one will work better. And sometimes it's bacteriostatic antibiotics. Because again, if you have a gram-negative organism that produces an endotoxin, then you want to inhibit that organism as opposed to destroying it. Because if you destroy it, then it will release that toxin into the body and the patient can get worse. Also, if you use bactericidal, it'll kill your normal microbiota. Um, Remember, antibiotics or medications, they have specific targets, but they don't differentiate between what we call good versus bad bacteria. The bacteria in our gut that help us digest food and break things down is good bacteria. We need them. Whereas, um, the pathogens, we want to kill them, but there's no way to differentiate. They can't say, okay, this one's good, this one's bad, good, bad, and separate them out. What they do is they look for a target, and if that cell has a target, they're going to work, they're going to do their job. Whether that's good or bad in terms of cell type, whether it's our normal microbiota or the pathogen, either way, the antibiotic is still going to do its job. So, antibiotics are naturally occurring in some cases. Penicillin, when Fleming discovered it, that's obviously going to be naturally occurring. Synthetic antimicrobials, these are manufactured by chemical process. And then semi-synthetic, they are modified, uh, modifying naturally occurring antibiotics. What's interesting is penicillin and ampicillin and amoxicillin, all the cillins, you would think they all have a similar structure, and they do. They actually have a very similar structure. But there are slight differences that are created when we use semi-synthetic antimicrobials. We modify them just ever so slightly, which means that they can be effective if you start to see resistance in the naturally occurring one. So for instance, penicillin started to see resistance. We went in, we modified penicillin just a little bit, just enough to make it look different to the bacteria, and the bacteria is less effective against fighting off that antibiotic. It's very neat how it works, and this class, unfortunately, we really don't have the time to go through all that, but just know antibiotics are different classes. There are different classes, and they attack different components. And we will talk about how and what they attack, and then some examples of each. Just maybe one example of each. They have to exhibit selective toxicity, meaning they can't they can't attack everything, right? We have to be very careful in terms of what antimicrobials we use. And yes, every medication you take, whether that's antibiotics or heart medication or whatever, there's always a warning list of contradictions and side effects. Not everything works the same for everybody, right? So penicillin may work for one person, but not another. It may, um, it just may not be effective, or they're allergic, so they have to use a different antibiotic. So what's important is we understand that yes, there is a slight risk, because if you have an allergic reaction to an antibiotic, that's obviously not a good thing, especially if it's an anaphylactic reaction, a really strong uh, response. So, yes, to a degree, we have to be careful, but we've been using these antibiotics for years and years and years. We're talking decades up to, centuries. Um, Almost a century for treatment. So the point is we have to be careful what we choose, what antibiotic we choose, because depending on what we're trying to treat, there are specific ones that are more effective and more efficient than others. Therapeutic index. So this is basically the maximum tolerated or safe dose versus the minimum effective, or what we call therapeutic dose. So maximum tolerated versus minimum effective. All right, and that tells us the therapeutic index. A high therapeutic index is considered safer than a drug with a narrow therapeutic index. So if the maximum tolerated, um, the ratio of maximum tolerated to minimum effective is higher, then it's going to be considered a little bit more safer than with something that's in terms of ratios pretty close to like 1 to 1 or 2 to 1. That's a bit more narrow for therapeutic index. So we want to be able to say, okay, we know that we can give this patient this much of this antibiotic and it will be effective. But you have to be really careful, because if you give too much of that antibiotic, it then in turn it becomes less effective for the patient and can make the patient even more sick by weakening their normal gastrointestinal or normal microbiota in general. So what we do is we want to figure out, okay, what's the concentration we can give that's high enough to be effective, but not go any further than that. Because if you go too much further, let's say it's at a specific concentration is 25 micrograms per mil, and that is the uh, that is the maximum safe dose versus that is effective. You don't want to go too far above that. If 25 micrograms per mil is the maximum tolerated safe dose and it's effective, then you want to use that dosage. But if you go too high, then you can do some serious damage to not only the normal microbiota, but you can also do serious damage to organs. You can have some organ issues, organ failure. So we'll talk about that in a second. So targets, and we're going to talk about each one of these. We'll talk about every single, all 5 of them, of the targets, and we'll give one example of each that I expect you to know. All right, so first off, cell wall inhibitors. Basically, cell wall inhibitors are really effective during the exponential growth phase, because remember, during exponential growth phase, you have lots of binary fission, right? And as a pathogen is undergoing binary fission, if you introduce a cell wall inhibiting antibiotic, then it's going to block that pathogen from creating or making the cell wall whole. And when it does that, it means the bacteria becomes more susceptible to antibiotics. A good example of that is penicillin. Penicillin inhibits the cell wall structure, um, inhibits cell wall structure. Basically, what it does is it prevents the synthesis of the peptidoglycan layer, which makes the cell wall weak, and the cell wall will eventually rupture and lyse. And again, it's more effective when the cells are undergoing binary fission, when they're growing and trying to create a new cell wall. If you have a mature cell that's not rebuilding its cell wall, penicillin's less effective because when you have a fully developed cell wall, right, when these bacterial cells have a fully developed cell wall, penicillin is not going to be able to inhibit the production of the peptidoglycan because it's already made. So sometimes that might not be the most effective antibiotic to use. Uh, Antifoliate drugs, basically these guys target folic acid production. Folic acid is useful, um, and when it comes to folic acid production, we can use sulfa drugs. So I do expect you to know that sulfa drugs target folic acid production, which is a B vitamin. Sulfa drugs are bacteriostatic and they are broad spectrum. Some antibiotics attack ribosomes, which inhibits what? Give you a second. If you're attacking a ribosome, you're inhibiting the ability to produce a protein, right? Because ribosomes are using translation to make a polypeptide. So some drugs can actually target the ribosome and prevent it from creating a functional protein. Now, when we talked about this, we talked about the fact that there are structurally two components to a ribosome, a large subunit and a small subunit. Now, depending upon the antibiotic you give, it will attack one subunit or the other. Now, we as humans don't have to worry too much about the fact that we also have ribosomes in our cells. Our cell ribosomes are very different than bacterial ribosomes. Okay, they're structurally different. So when we give an antibiotic that attacks ribosomes and protein synthesis, then our bodies, our human cells, don't have to worry too much because they're very different. Now, there is a catch though. Within the human cell, though, where else do we find a ribosome that's similar to a bacterial ribosome? It's going to be the mitochondria, right? The mitochondria, based on the endosymbiont theory, is used to be its own individual prokaryotic cell. So its ribosome is very similar to bacterial ribosomes, almost identical. So it could pose a problem to mitochondrial ribosomes, but human ribosomes in general are not really affected by this because they are very different in structure and these antibiotics target bacterial ribosomes. Now, there are a lot of examples: macrolides, lincoamines, chloramphenicol, which you guys used in the lab, tetracyclines, aminoglycosides. The one I really care that you know, and it's just good enough, is tetracyclines. So tetracycline antibiotic is going to prevent protein synthesis. Some antibiotics target membrane structures, meaning they make the membrane more permeable. They make the membrane more permeable, specifically the plasma membrane. A good example of that is polymyxin B and colistin. Now, I only expect you to know one of them, and that's polymyxin B. And basically what it does is it triggers changes in the permeability of the plasma membrane. And when the permeability changes, things leak out of the cell, and the cell will eventually die. This is a more narrow spectrum antibiotic, meaning you give it for very specific pathogens. But point is they target the permeability of the cell membrane, and by doing that, they trigger the loss of important metabolites, and eventually the cell will lyse because the membrane is too weak to hold it together. I expect you to know polymyxin B from these two. Targets for viral replication. So we haven't really talked about viruses too much in this class, and we haven't really mentioned viral replication at all, which is fine. This is just, I don't expect you to know this slide. I just want to show you that antivirals are classified by the activity. So attachment, penetration, encoding, replication, and then release. So antivirals also have these mechanisms that they use at certain points, right? You're going to have a very specific antiviral that attacks uncoding, and one that attacks replication. So they're also pretty specific like antibiotics. But again, we didn't go through all this in any previous chapters, so I don't expect you to know it. Susceptibility. So this is something you guys should have seen before because you did Lab 7. And Lab 7 was talking about the susceptibility of an organism to a given antibiotic at a given concentration. We talked about therapeutic index and how we want to figure out how high of a dosage can we give that's safe but also effective. Okay, so we want to figure out how high of a dosage can we give while still making it safe and effective. And one of the ways to do that is to determine the MIC, the minimum inhibitory concentration, the minimum amount you can give to inhibit that pathogen. And we do this through susceptibility testing, and you guys should know what that is, considering you just did it in Lab 7. Okay, you actually determine susceptibility using the simulation and the actual lab itself. Let's take a look at disk diffusion. So you've used disk diffusion. Well, actually, you've seen it. I created the plates for you. I put down the disks, and then we saw what would or wouldn't grow around that disk. So for Lab 7, what I did was I used a pure culture of E. coli, and I plated it, and then in the middle of the plate, now you can see in this example, they used lots of discs. But in the middle of the plate, I used chloramphenicol for one, and then the other one I used ampicillin. And what I did was I let it grow, gave it a couple days in the incubator, and saw what happened afterwards. again, we use Mueller-Hinton auger in this case, and what we do is we look for a zone of inhibition. So the zone of inhibition is the area that spans outside the disc that you see no growth. So here's the disc, all the way to here, all the way to there, and everything around that. That's your zone of inhibition. It is inhibiting bacteria from growing. So whatever this antibiotic is, and whatever the concentration is, then it's extremely, or well, it depends. It's effective depending upon what pathogen you're looking at. Larger the zone of inhibition, you can usually kind of make the inference that the more effective it is, or the larger the zone of inhibition, the more effective it is. Um, Then if you move over to something like this one right here or this one here, whatever antibiotic this is at whatever concentration, it doesn't matter. The bacteria grew right over it. Meaning if you gave the patient this antibiotic at this concentration, it will literally do nothing to stop the bacteria. It's not a high enough dose or the correct antibiotic. What you want to do is you want to find the antibiotic that has the biggest zone of inhibition and say, okay, well, this medication at this concentration is pretty efficient because look at the size of the zone of inhibition. Now, when it comes to the zone of inhibition, there aren't arbitrary numbers. We actually have a book called an antibiogram book that's several hundred pages long, and it gives us all of the different combinations. So if you're growing for instance, you're growing E. coli, you use ampicillin at 10 micrograms per milliliter on your disc, and you measure the zone of inhibition, and it's 9 millimeters. Well, in the antibiogram book, it tells you, okay, under this many millimeters, we still consider it resistant. If it's between this one and this one, it's moderately susceptible, meaning the bacteria is moderately susceptible to the antibiotic. Or if it's above this number, the bacteria is extremely susceptible to this antibiotic at this concentration. So it's important to read and calculate what the zones of inhibition are, because we can then say, all right, well, based on this antibiotic at this concentration against this pathogen, it is this effective. So there's a lot of things that need to go into all of this. And we're determining the minimum inhibitory concentration, the minimum amount we can use to inhibit the bacterial growth. Okay. Hopefully this is kind of clear. If you have questions, see me in office hours via Zoom, and I'll help with this a little more. Another way to do this is an e-test. I'm going to skip that slide because you can read it, and I'm just going to show you what it looks like. We're still using, so the first one was disc diffusion. This one is called the e-test or the epsilon test. And basically what it does is it does almost the exact same thing as the disc diffusion test, except it has varying concentrations through the whole test strip. So, for instance, this test strip has VA, which is vancomycin, that's the antibiotic, and right here at this point is the concentration. And this is the concentration. And the concentration goes up as you go up the e-test. The higher the concentration as you go upward. Now, what you're looking for is the minimum amount that you can give, yet still being effective. So when you look at these, you want to look to see where there is separation from the actual strip. So if you look at 2.25, where this line is drawn, there's separation right here. This tannish color right here, this is all growth. the clearing means there's no growth. So if we look right about here, there's no growth on this side, and I know the arrow's a little bit in the way, but there's also no growth on this side. So what you're looking for is when the growth separates from the actual test strip. So our minimum inhibitory concentration for this would be 0.25 micrograms per milliliter. If growth continued, and let's say it was attached, there was all growth in here and here up to about here, right? We would say if wherever the separation is, the MIC, the mic, is that concentration in milligrams per microliter. So similar concept to disk diffusion, just a little bit different in how it actually functions. But you still have different concentrations for this antibiotic, in this case vancomycin, and you look for the separation where the bacterial growth is not touching the test strip anymore. And when it's not touching the test strip anymore, that tells you where the susceptibility is, or what the susceptibility is. Superinfections. So superinfections result when superbugs become primary bacteria in an infection. Now, we have quite a few superbugs in the US and across the globe, and we're starting to see more and more over the last decade. Basically, this shows us how superinfections develop. All of a sudden, in this first one, some of the microbes become resistant. The susceptible cells, they die because they're not resistant to, excuse me, to the antimicrobial drug. And then the population of the resistant pathogens, well, since they're some of the only ones left, that number is going to increase. Right, that number is going to go up, and then all of a sudden you have a population that's all resistant to antibiotics. And we actually talked about this early in the semester. We mentioned that evolution through natural selection and how introducing an antibiotic, if you don't take the whole thing, if it's the wrong concentration, it can cause these bacteria to develop resistance. Then, since they're resistant, they're going to outcompete the ones that will die because of the antibiotic, and they become the majority in the population, which then becomes a problem for you because now you have a superbug that is resistant to almost all known antibiotics, if not all, and there's really, in some cases, no way to treat it. Then, if it's there, it can potentially transfer its genetic material to another cell, and that cell will also become resistant. That cell will also become resistant, and we've actually talked about some of these mechanisms. So we're going to talk about intrinsic versus acquired resistance. Now, I'm not going to give you the definition just yet because I have an image on the next slide. So the two types of antimicrobial resistance are intrinsic, which natural, and acquired means you get it from somewhere else, or the bacteria gets it from somewhere else. This image is really good. I don't have the number here, I apologize, but this is a really great image. It shows you antimicrobial resistance, intrinsic, which is natural. For instance, endospores, capsules, cell wall composition, all of these are intrinsic examples of ways bacteria can resist antimicrobial medications. Then you have acquired. Acquired, you could have mutations. A simple mutation could lead to resistance. or you could have horizontal gene transfer. And we absolutely talked about all three of these methods. Conjugation, transformation, and transduction. Conjugation, remember, they create the bridge, the sex pilus, and they transmit genetic material that way. Transformation, they can pick up genetic material from the environment. Or transduction, a bacteriophage will accidentally transmit some bacterial DNA into the new bacterial host cell. So there are three major ways through horizontal gene transfer, and then you have mutations as well for acquired, and then for intrinsic, you're talking about things that these cells already have that make them resistant, and that includes the cell walls, their structures, capsules, slime layers, endospores, among many other things. Okay, so intrinsic is natural, acquired you get it from the bacterial cells get the resistance from somewhere else. So I'm skipping through these slides because we've talked about them. Definitely read them, but I'm not going to spend time talking about them because we did go over them several times. Now, how do microbes evade antimicrobial drugs? We talked about how antimicrobial drugs attack the pathogen, but we haven't talked about how bacteria evade those antimicrobial drugs. And three major ways are altering the target, inactivating the drug, and reducing the concentration in the cell. So let's go through one by one. Here is your, let's say this is an enzyme right here. Normally it would fit this drug, it would bind to this target site, and the antimicrobial drug would go to work. But notice they developed, right here, this little spike in the middle, a change in the target site, and when this antimicrobial comes to bind, it bounces off that middle spike because it can no longer fit. So changing the target site just a little, not even a lot, just a little, is enough to prevent the antimicrobial drug from binding. Drug inactivation. There are some really good examples of this for penicillin, for instance. Penicillin has the ability to break down the cell wall, right? But they have a very specific structure. They have what we call beta-lactam rings. So in their chemical structure, they have rings. There's several of them. And bacteria can have something called beta-lactamase, which is an enzyme that will chop those rings up. So if the penicillin enters and that cell has beta-lactamase, it'll go right up to the rings in the structure of for the penicillin, and it will chop those rings up, and the penicillin is no longer effective. It's no longer effective. So they can actually literally destroy the antimicrobial drug that's entered into the cell. And then there's two other ways to reduce concentration. Reducing permeability. We know that cell walls and cell membranes have a certain amount of permeability to them. But if the cell starts to reduce that permeability, less and less of the antimicrobial will be able to enter into the cell. So that's a good thing for bacteria because it can prevent the concentration from getting high enough to be effective. And that's kind of what this last one does right here, an efflux pump. An efflux pump. I find these very interesting because basically what it is as the antibiotic or the antimicrobial medication enters the cell, the cell pumps it back out, and more enters and it pumps more out. So what it does in theory is it never lets the concentration of the antibiotic get high enough to be effective. Remember, for like the mic, we need the minimum amount that's still effective, right? So if the bacteria can keep that concentration below that number, then the antibiotic won't be effective. It's kind of like, um, you have you're in a little boat, little canoe, and you have a leak, and you are paling out the water, right? As long as you can pail the water out faster than it can fill the canoe or boat, then you'll have enough time to make it to shore. For bacteria, as long as they can pump the antibiotics out as fast as they can as it enters to keep the concentration low, then they will survive longer because the concentration never gets high enough to kill them. These are all really great ways of cells, of bacterial cells, um, blocking or preventing antimicrobials from being effective. And this is just a list: altering targets, inactivation, efflux pump, and then limiting permeability. So non-compliance is the biggest reason why we start to see antibiotic resistance, and that's because patients, it's patient misuse, right? They miss doses, don't finish the prescribed amount of time, and then self-medicate with leftover prescriptions. So those are really common. People feel better. Let's say they're prescribed a 10-day course. They feel better after 5 days, and they say, okay, I'm going to save these remaining 5 days for the next time I feel like I'm sick with this, with whatever this infection is. And they wait and they wait and they wait, and I have them set aside. Well, as they wait, the the efficacy of that medication starts to decrease. It starts to degrade, so it becomes um, less effective and efficient. Excuse me. Now, misuse is hopefully we can manage it on our own. If you are given a prescription for 10 days, take all 10 days. If you're given a prescription for a very specific antibiotic, take that antibiotic. Make sure it's the right concentration. Um, because if you introduce what we call a low dose pressure, meaning introducing a low concentration of the antibiotic into your body, then what you're doing is you're setting yourself up to develop resistant bacteria within your body. Right? And we talked about that early in the semester, how selective pressures, right, will lead to resistant traits. So low dose antibiotics, low dose exposure, you definitely want to um, avoid that. You definitely want to avoid that. Uh, Microbes to watch. CDC has a whole list of um, microbes that we need to pay attention to. We break them down into urgent and serious, and this is referring to resistance. Urgent, as of 2017, C. diff, Clostridium difficile. We'll talk about that later in the semester. Extremely contagious, extremely dangerous. CRE, again, extremely resistant. And then now we're starting to see even more so drug-resistant gonorrhea, which there's a couple cases in the U.S. There have been a couple cases in the U.S. over the last 3 or 4 years where patients have this drug-resistant version of gonorrhea, and there's really nothing we can do because it's resistant to almost all known antibiotics. Serious concerns, we're starting to see multi-drug resistant strains of all of these organisms. Uh, Staph and strep and Pseudomonas and chlamydia, or excuse me, candida, not chlamydia, candida. Uh, so candida is actually a yeast, but that's okay. It's still obviously a microbe. But Pseudomonas, Mycobacterium, Staph, Strep, Campylobacter, those are all bacteria, where candida is a yeast. Again, we're starting to see multi-drug resistant strains of these organisms. I don't expect you to memorize them. I just want you to see that by allowing misuse of antibiotics, we're starting to see really dangerous repercussions. And this image right here makes a lot of sense. Hopefully you can see how whatever is living on this plate is extremely susceptible to this antibiotic. It's extremely susceptible to this antibiotic. 10 years later, the bacteria is extremely resistant to that antibiotic. It's definitely not something that we should be trying to take part in. We want to our limit our responsibility in terms of antibiotic resistance, right? We want to follow the prescriptions. We want to make sure that we don't misuse them, because what we're doing is we're just doing ourselves a disservice as well as other people, because now we start to see resistant strains of other pathogens, and maybe our body can fight them off pretty well, or at least moderately well, and some other people's bodies, if they're immunocompromised, can't. So it does rely on us to take that step and make sure that we're using antibiotics properly to avoid this resistance. Now, that's the last slide. I just want to go back to one of the other slides first. And I want to talk about the ones you need to know. So from this slide right here, okay, I'm telling you exactly what you need to know. So for cell wall synthesis, that's the target, and the example is penicillins. Plasma membrane, polypeptide drugs, which is polymyxin B. Polymyxin B for plasma membrane. Target, nucleic acids, quinolones. So for nucleic acids, I expect you to know quinolones. For protein synthesis target, tetracyclines. For protein synthesis, tetracyclines. And for folic acid synthesis, sulfa drugs. Sulfa drugs. So, cell wall synthesis, penicillins. Plasma membrane, polymyxin B. Nucleic acids, quinolones. Protein synthesis, tetracyclines. Folic acid synthesis, sulfa drugs. I want to make sure that it's crystal clear. Those are the ones I expect you to know for examples of each target within or antibiotic target. Okay, that is the end of this chapter. Everyone stay safe, and I will speak with you all soon. All right, thank you, everybody.